Hello, people. So I hope it is okay with you that I wanted to wait till 3:05. Although if nobody will be, no other students will be joining, I'll maybe start sooner, maybe in a minute. And okay, so I actually have a lot to cover today. And anyway, there are recordings of these sessions, so most of you might be joining on those uh, recorded lessons. So maybe I'll start earlier today. I'll wait for just another minute. If at all any of your colleagues want to join the live session. Okay, maybe I'll start with the meeting because anyway there are recorded lectures that are available, recorded meetings. These sessions are recorded, and anyway I'll be providing you with those sessions later on. So I'll start for now. If in case people are joining, I'll just hold for a while and let them in. So for that purpose, let me just share my screen. So what we are going to do today is we have basically tackled the previous year's assignment for the first and second week. My aim today is to tackle the third and fourth week's previous year assignment as well as the extra concepts that need to be clarified in week 3. All right. So next week for our last lesson, we'll just be left with week four's extra concepts. And what I'm planning to do in that session is also discuss this year's assignments because by then all the solutions would have been out, right? So I'll discuss this year's assignments plus week four's concepts. Plus, if at all you want to do any extra practice problems, for example, those vector problems or pedigree problems, if at all you want to do practice for those particular things, so maybe I can just add up some questions and maybe we can tackle them also in our final week, right? So, in order to be able to do that, since we just have 2 hours, in the final week also, I will be tackling with both third week's and fourth week's previous year's assignment this today itself. And we'll do our concept clearing also later on. Right. So in order to do that, I'll be sharing my screen. If at all people will be joining, I'll just halt and let them in. And you don't have to be very vocal throughout the session, but if at all you are unable to see my screen at any point, please do inform me because I have no other source of information regarding that, right? So if my screen at any point is not visible to any of you guys, please just stop me then and there and inform me. So for now, I'll be sharing the screen. Okay. I hope it is visible. Just a moment. Just a moment. I just stopped sharing for a moment. I think it is visible, right? So I'll just put it on the slideshow mode and I'll continue dealing with this. I have no clue why my video is not being presented along with this. Okay, anyway, that is not a big concern. So I'll just move on with this assignment. Let us first talk about the previous year's assignment for week three. Week three was basically talking about molecular pathology, right? So we are just going to see these questions. I am going to give you a little bit of time to think about these questions, and then I'll be showing you the answer. And wherever discussion regarding the answer is required, we'll be discussing that, right? So the first question here is PCR. Just a moment, let me just get the pointer right. PCR can be used for which one of the following processes? A. Cloning a segment of the non-coding region of the chromosome. B. Cloning coding region present in the processed mRNA. C. Mutation detection. Or D. All of these. So PCR is something that as a technique hasn't been extensively discussed, but with respect to what has been asked in this question, it has been discussed ample amount of times in sir's lectures itself. So I hope you people know the answer. Let me just show it to you. So the answer is all of these. So PCR, what is PCR? It is polymerase chain reaction. What is the major principle of PCR, or what is the major function that it performs? It will basically amplify whatever a sample is given to it. It will be amplified, right? So this amplification is not limited to just the coding region or non-coding coding region, right? Either of these could be done by a PCR if the appropriate primer has been used, right? Apart from these, mutations are detected using PCR. One such example is a very common ongoing this pandemic test, which is RT-PCR basically, which is a variant of PCR, which is basically doing what? It is detecting a mutation in that virus, right? Or it is detecting the presence of that virus, right? So this okay, I deviated from the topic a bit. That is basically detecting the presence of the virus, but mutations in our genome can also be detected using a PCR in two ways, basically. One, a particular sample, if it is in a very small quantity, it can be amplified up to that number, that mutation basically becomes more visible, right? The other aspect where a PCR can be used to detect a mutation is that, for example, a mutation is such that it basically hinders the process of this amplification or DNA replication in general using polymerase. So if that process will be hampered, so the PCR products will not be able to be formed. So we know that, okay, this was the gene or the coding or non-coding region of the genome that we were targeting. And since it could not be amplified, maybe there is a mutation in this region, right? So there are multiple explanations. So I have also given you illustrations of how basically PCR is involved in these steps that we talked about. So in cloning, how is it used? Basically, PCR will be used to amplify the genome sequence that we have to basically clone, right? So the cloning vector could also be amplified by PCR, and thereafter, once this vector is actually containing whatever genome we also require, and it is in the form of a plasmid, and once it is injected into the bacteria or E. coli, for example, then E. coli's replication machinery itself will take care of the rest of the amplification or multiplication process, right? But up till this, to at least amplify the cloning vector, PCR can be used. And this can contain either the coding or the non-coding region, right? Apart from this, use of PCR in detection of mutations, like I just talked about, this is just an example that I have picked from Abnova's site, right? Over here, what they say is that suppose there was a DNA polymerase. So what will a DNA polymerase do? It will basically polymerize a new DNA chain, or it will synthesize DNA, right? So if for example, this EGFR is a gene. If the EGFR was a wild type EGFR, Then basically, and we also added this PNA probe over here, which is a perfectly matched probe for wild type. Okay, so if we know that this gene has no mutation in it, it is a wild type gene, this probe will basically show perfect complementarity to this gene. And since it will bind on the region where this gene is present, DNA polymerase will not be able to proceed further. It will basically get dislodged from that sequence and new amplicons will be formed, right? On the contrary, if this EGFR had a mutation in it, right, then this PNA probe, which was supposed to be perfect complementary to the wild type EGFR, will not show perfect complementarity to this mutant EGFR, right? And it will not bind to this this gene region, due to which the DNA polymerase will not get dislodged. Instead, it will actually synthesize the entire chain, which will basically give amplicons, which is how we will judge for this particular gene whether there is a mutation or not. So, such examples are also in line of how PCR is basically used in detection of mutations. So sorry. So, moving on to the next question. Which one of the following options correctly identifies the process given in the schematic diagram? So the schematic diagram basically shows a pre-mRNA, right? It has different exons which are colored differently, and it is showing some kind of a process which is splicing, right? Which is mentioned over here. Then splicing is yielding some kind of products which after translation, or what does translation do? It basically reads the mRNA code and converts that code into a polypeptide or a protein, right? So after translation, there are certain protein isoforms that are formed, right? So you have to basically identify that which of the options that are given basically correctly describes this process, right? So your options are trans splicing, alternative splicing, self splicing, or tRNA splicing. So which one of these is basically the process that is illustrated by this particular diagram? You have to basically uh, come up with the answer. I will also let you know the answer. And let me give you a hint regarding that, that proteins are different, right? Protein isoforms means that there are two different types of proteins that are formed, right? So proteins over here are different. This is the key giveaway of what kind of process this will be, right? So coming on to the answer is basically alternative slicing. So this is probably the most, you know, uh, the term that you might have heard the most up till now. So alternative splicing it is. Alternative splicing basically means that from a single pre-mRNA or from a single gene sequence, which will basically give this pre-mRNA, there will be different kinds of functional mRNA that will be produced basically, right? I just need a moment. Huh. So there will be different types of functional mRNA that will produce, which will form different functional proteins, right? This process will basically be known as alternative splicing. For example, in this first functional mRNA, just a moment. Huh. For example, in this first functional mRNA, exon 1, exon 2, and exon 4 have been basically spliced together to form a protein A. And in the second one, exon 1, 3, and 4 have been spliced together to form this protein B. Right? So this process is known as alternative splicing. Now, I also wanted you people to understand what this trans splicing and self splicing actually is. Right? So trans and self. Okay, self-splicing is also known as cis-splicing. Okay, this is the first thing you need to know. In most of the literature, you will not find the term self-splicing. Instead, what you will find is cis-splicing, right? So they are both just synonyms. So over here, let me explain you what cis and transplicing is, or what self and transplicing is. So we have read it in other contexts, what particularly chemistry and biochemistry, what exactly cis and trans mean, right? So if anything is on the same plane or on the same side, it is referred to as cis, and if it is on opposite sides, it is referred to as trans, right? So similarly, here in biology also, cis splicing basically means that uh, uh, two exons on the same particular mRNA will be spliced together. And how is it different from alternative splicing is basically that the intronic region over here will basically be self-sufficient to carry on the splicing process, right? So this particular intronic region that you can see here in this peach color, right? This will basically act as a self enzymatic, it will have some self enzymatic activity that will basically splice this exon 1 to this exon 2. So since they are on the same particular mRNA, it is known as cis splicing. And how is it different from alternative splicing? Because this particular region itself, without the help of any other enzyme, it is performing the splicing, right? And what is trans splicing? So, like trans, the sites are present at two different locations, right? Over here, it will be at two different RNAs, right? So, the first exon is on this sLRNA and the second exon is on this pre-mRNA. So, this is just a representation and example, right? So, what you need to understand is that this first exon will be spliced with the second exon, right? Which are present on two different RNAs. So that is what to form a mature mRNA, right? So that is what this trans splicing is. What is tRNA splicing? It is similar to this kind of splicing, but if it would have been a tRNA splicing, protein would not have been formed. Okay, because it would not have been a protein coding region. It would have been a protein non-coding region because it is forming tRNAs and not functional proteins, right? So moving on to question number 3. Given below are two DNA sequences and their corresponding transcribed amino acid sequences. One for the wild type allele and the other for the mutant allele. So this is a wild type and this is the mutant allele. The mutant allele shows one base pair change which is highlighted in red and converts amino acid cysteine to in the wild type polypeptide chain to serine in the mutant allele. A mutation of this type is known as which one of these? Missense mutation, nonsense mutation, translocation mutation, or insertion mutation. So please think about it for a moment. Just let me have water. Also, I think there are some people in the lobby. Let me just admit these people. Okay. Okay. So I have added your colleagues. Let me just to be sure, let me stop sharing and I'll share the screen again so that there's no problem with the presentation, right? Also, people who have just joined, what we were actually doing was going through the week three's previous year assignment. After this, what we'll be doing is going through week four's previous year assignment, plus we'll do week three's concepts that need to be cleared, right? So let me share my screen again. Okay. So I hope it is visible to all. This was the question that I just read. So time for its solution, right? So this kind of a mutation is basically a missense mutation. Just let me get a pointer. This is a missense mutation, right? So it is clearly visible that one amino acid basically gets changed to other amino acid, right? So it has bound to be a missense mutation, which basically means that because of the change of one nucleotide or one base, the basically amino acid that was being formed was changed, right? On the contrary, what are these other mutations? I think I might have put in a slide. Yeah, I have. So first to tell about this particular mutation, which is translocation mutation. So there are two basic classes of mutations, right? One class includes these mutations which are missense, nonsense, or silent mutations. The other ones include chromosomal which are known as chromosomal aberrations. They basically include these mutations which are duplication, which basically means that one genomic region is basically duplicated, right? The other is inversion. For example, if this particular place was A, okay, I need a pen. If this particular place was A and this was B, now this is B and this is A. So the sequence has been basically inverted, right? Apart from this deletion, as you can see, this particular region has been deleted and this is the product. In insertion, for example, this is one chromosome, this is the other chromosome, and a part of this chromosome, in this particular case, a part of this chromosome has been transferred to this chromosome. So there is an insertion over here and there is a deletion over here, right? So insertion over here and deletion over here, the other way around could also have been possible. And then coming on to translocation, what is translocation? That basically, uh, actually, there are two kinds of translocations. It could either mean that one part or one region has basically moved on to this part, right? It would kind of be like an insertion. And what is shown over here is a reciprocal translocation, that there are two chromosomes that are involved. A region of this chromosome has come onto this chromosome and vice versa. A major chunk from this chromosome has moved on to this particular chromosome, right? So these are chromosomal aberrations. Now, apart from this, let me get the pointer back. The other kind of mutations, this is something that I have already discussed, right? So the mutations that were mentioned over there were missense and nonsense mutations, right? So in case of missense, what happens is that the amino acid is basically switched to another kind of amino acid. For example, this particular codon was coding for leucine earlier. Now, because of mutation in the first position of this codon, it is coding for valine, right? But on the contrary, what is a nonsense mutation? A nonsense mutation basically means that earlier the codon was coding for a protein, but due to some change in its base, what happened was that now it will act as a stop codon, right? It will not be a protein coding codon. It will just be a stop codon. So this particular case can be seen if there was a mutation in this second base, right? And this is the result of that. Then what would be a silent mutation? So as we had basically, when we had discussed the genetic code, we had seen that there is some redundancy in genetic codes. So for some particular amino acids, there are more than one codons that can basically code them, right? So for leucine, UAA can also code leucine, but if, for example, there was a mutation and a cytosine was added over here instead of uracil, then still it would be coding for leucine. So that would be a silent mutation, right? And the other option over here was basically insertion mutation. So if there is an insertion, like we talked about here, then the reading frame of this particular codon will change, right? So if there is an insertion over here, it will basically be a frame shift mutation, and the following amino acid sequence will entirely change. So that is not really happening over here. So over here, the answer was missense mutation, right? So moving on to our next question, and let me just add the people who are waiting in the lobby. So I've admitted, I hope my screen is still working. Oh, it's not working for me only. It's working. Okay. So question number 4 says, you plan to develop a microsatellite marker from a genomic locus bearing a polymorphic repeat of CGG triplets, right? The sequence surrounding the triplet region is made available to you, and the same is shown in the schematic diagram given below. Which one of the following options identifies the correct forward primer and reverse primer respectively, which as a pair can be used to amplify the repeat region? Okay, so this is a trinucleotide repeat that is given. Hannah? So what these people are saying in the question is that the flanking region sequence has been made available to you. Depending on that, you have to basically design a forward primer and a reverse primer. And I will give you some time to just look at this question to see if you can come up with the correct answer. Otherwise, I'm obviously going to disclose the answer to you, right? So please take a moment, look at this question. It is a good question. It might help you in your finals for this particular course. Meanwhile, I'll just have something for my throat. Since it is a little big, right, the questions and the options, so I'm giving you a little extra time. Also, somebody is waiting in the lobby. Okay. All right. So I'll give you 30 more seconds maybe to just look at this question, try to come up with its answer, and then I'll be discussing the solution. Okay. So coming on to the solution for this question. Okay, I'm unable to switch slides. So this fourth one is the correct option basically. So over here, what we had to do was we had to design a forward primer and a reverse primer. So since the sequence in the middle is the same, right? It is a trinucleotide or polymorphic repeat of CGG, right? So one hint was that one strand basically had this in forward direction, the other had it in a reverse direction. Why? Because okay, and we need to basically keep in mind certain rules of uh, basically DNA synthesis, which I have mentioned over here, that the forward primer will basically prime from the left end, right? And in the 5 dash to 3 dash direction, right? Whereas the reverse primer is a reverse complementary to DNA sequence and thus primes leftward from the right end, right? So DNA synthesis will always take place in the 5 prime to 3 prime direction, right? Depending on that, this will be our sense strand. Sorry, this will be our anti-sense strand and this will be our sense strand, right? So from our anti-sense strand, basically an mRNA is synthesized. So over here, a forward primer will attach. So the forward primer will have to be complementary to this particular region, right? So is this forward primer complementary to this particular region? It says C, then A, then G, then T, and maybe you can match the rest. So it is complementary to this particular region, right? So, since DNA synthesis is anyway going to take place in this direction, which is the forward direction for the synthesis to occur, the forward primer will be complementary to the sequence. Now, keeping this in mind, the reverse primer will have to be complementary to which particular sequence? It will basically be complementary to this sequence, right? But over here, also the synthesis will take place in 5 prime to 3 prime direction, right? So 3 prime is over here. It will start over here, right? So you basically have to invert this sequence and see whether it is complementary or or the reverse primer will be directly complementary over here, right? These are basically palindromes. So it is whatever is given over here is just the reverse is given over here, right? So it will basically, if you want to look at it in the same orientation, you can basically match it over here. No, you will have to match it over here only. So is this particular sequence, which is in 5 prime to 3 prime direction, complementary to this particular sequence? Let us check. So A, T, T, then ACG, ACG, CAT. Right? So it is. So that is why this is the correct answer. It might have been a little confusing for you if you were not aware about what forward and reverse primer does. But for that, you can keep the statement in mind. This was the statement that was basically mentioned in the previous year's solution, right? So the forward primer, what will it do? It will prime from the left end in the 5' to 3' direction, whereas reverse primer will basically be complementary to DNA sequence and thus prime leftward, right? It will prime like this from the right end. So, eventually you will plan your PCR experiments. You will have to design primers for that. This particular information will come handy, right? When you are actually performing your experiment. So moving on to question number 5. Okay. A steroid hormone receptor X, upon binding to the steroid, activates a set of genes involved in the physiological pathway promoting muscle growth. Due to the genetic mutation, I think there's somebody who's trying to join. So since we have not really started the question, let us just admit that person. Okay. I hope I was able to admit the people who were not present. And I resume my screen share, right? So let me begin the question again. A steroid hormone receptor X, upon binding to a steroid, activates a set of genes involved in the physiological pathway promoting muscle growth. Due to genetic mutation, just a moment, due to a genetic mutation, the receptor X was able to interact with a few novel targets and activate their signaling cascade, right? Which one of the following option correctly identifies the type of mutations involved? So the options basically are dominant mutation, dominant negative mutation, gain of function mutation, or missense mutation. I don't know why my pen did that. So which one of these mutations is basically talked about in this particular scenario? I think it is a relatively easy question, unlike the previous few questions. So let me just jump to the solution. So it is basically going to be a gain of function mutation. So why is it a gain of function? Because it clearly states that due to this particular genetic mutation that is being talked about, this receptor X was able to interact with a few novel targets, which means that earlier this receptor X would not have reacted with these targets, but due to whatever genetic mutation it underwent, it basically increased its function to basically interact with new targets as well. And it could activate their signaling cascade, which is why this is a gain of function mutation, right? So moving on to question number 6, which says a de novo mutation can be either somatic or germline. Which one of the following statement correctly differentiates between somatic and germline mutations? So this is a very fundamental concept, right? So we are just going to read the options that are given and jump to the solution. The first option says a somatic mutation is heritable, whereas a germline mutation cannot be inherited. The second option says a somatic mutation cannot be inherited, whereas a germline mutation can be inherited. The third option says a somatic mutation occurs in a DNA sequence, whereas a germline mutation takes place in an mRNA sequence. And the final option is a somatic mutation is deleterious, whereas a germline mutation is non-deleterious. So, since it is another easy question, we'll just jump to the solution. I hope that you are already aware of what the answer is, but we'll see that the answer is a somatic mutation cannot be inherited. That's why it is basically, this is a fundamental difference between somatic and germline mutations. That germline is basically going to be a mutation in your germ cells, that is in your ova or your sperm, right? So in your egg or sperm, if there's a mutation, it will obviously be inherited. But in your somatic cells, for example, your muscle cell, your cardiac cell, if there is a mutation that does not move on to your germline, right, or to your egg or sperm, it will basically not be inherited, right? It will remain in your somatic cell. For example, a de novo skin mutation or a de novo mutation giving you some heart condition, right? It might not be inherited if it is not in your germ cells. So, again, I have just shown an illustration to basically prove this concept that germline mutation, it will basically be present in your sperm or ova. And because it is already present in one of these, it will be basically present in the embryo and in the entire organism, right? Because the fusion of these germ cells will basically form this particular embryo, and each and every cell of this embryo will carry this mutation, right? So the entire organism will have this mutation. And when we come to its gametes, it is actually not always uh, this case that half the gametes will carry the mutation and half won't, but it is also possible that maybe all the gametes carry the mutation, right? Whereas in somatic mutations, what happens is that there are normal germ cells or parental gametes. Then there is an embryo, maybe due to some mutagen exposure that this embryo might have gone through. There was a somatic mutation, right? And that somatic mutation will only be carried on to some part of the organism, which will basically not be expressed in all the cells, right? So this particular part might only be expressed in the upper thorax region, right? So only the upper thorax region will basically present that mutation, and it will not be carried forward to the progeny. So question number 7 over here says, given below is an agarose gel run after sequencing a DNA fragment by using Sanger sequencing method, right? Which one of the following options correctly identifies the sequence being generated? So I hope you are already aware of how to solve such questions. One such question I think was also present in your this year's assignment, right? Because I got a query for that also. So, just take a look at it. Maybe I'll give you 15 seconds to take a look and decipher which one of this is actually going to be the answers, which one of these options, and then we'll move to the solution. Okay. So I hope you already figured out which one the answer is going to be, and we'll just move towards it. So, okay, why I was also emphasizing about less time was basically because all these others started with T, right? So there are two things that you have to keep in mind over here. One, the DNA fragment will always be in a 5 prime to 3 prime direction, right? So in 5 prime to 3 prime, you always read it from this way to from hair to hair, right? So the first strand over here is A, the second is C, the third is G, then there are triple C. Is that what is given over here? Yes, that is what is given over here. Another possible option could have been C, because from 5 prime to 3 prime end, it still says A and C and G and triple C. Up till here, it is actually perfectly correct, right? Then you will have to actually tally the entire sequence to find out which one of these is going to be the correct answer. And this is the more favored orientation, 5 prime to 3 prime. So that is why we are selecting this particular option, right? So there could have been a confusion between C and D, but on what basis will you basically select D? Because the new strand or the basically outcome of Sanger sequencing will always come in a 5 prime to 3 prime direction, right? So moving forward to question number 8. Mutation in a gene causes a severe neurological disorder in often in the offspring of both sexes, but only when inherited through the mother and not when transmitted through the father. This property of the mutation is likely due to which one of these? Complementation, genomic imprinting, uniparental disomy, or anticipation. So these are basically concepts that I have discussed extensively. I hope that people who have followed those discussions would be aware of the answer. I also hope that people who anyway know about the answer, it might be correct. I'll move on to the solution, right? So the solution for this is genomic imprinting, right? What is genomic imprinting? It basically, first let me read this out. A few genes have the property to exhibit allele-specific expression, right? It is an intrinsic properties of some human genes that is there. That is that the allele will only express if it is either from the maternal or paternal allele, right? This process is known as genomic imprinting. Here, gene in question is most likely silenced in the paternal allele, and hence defective maternal allele leads to disorder, right? So we have discussed this, that what this basically means, allele specific expression, is that like the random X inactivation in females, where out of two X chromosomes, one is randomly inactivated, there is another such process that a few genes, they will basically show this allele specific expression such that if it is a paternal allele, it will directly be silenced and only the maternal allele will be expressed. So, if this maternal allele is basically defective, it will cause a disease. On the contrary, it is also possible that certain genes have the maternal allele naturally silenced and the paternal allele will basically function, right? But if this paternal allele is defective, then again, it will lead to a disease condition. So that is what genomic imprinting is. So moving on. Um, just hold on. Give me a break. Okay, I'm unable to turn this off also. I have no. Okay, I will share my screen again. I'm sorry for the interruption. I've had this ongoing illness for the past 1 month, I think. Uh, just a moment. Why is it not showing? Okay, I hope my screen is visible. If not, please inform me. Okay. So, these terms that were mentioned over here in this particular question, why am I need to switch? Huh. I wanted to actually discuss these terms also, which were complementation, genomic imprinting, uniparental disomy, and anticipation, right? So that you people are clear with what all this is. And if at all somebody asks you something other than genomic imprinting, also, you might have already read it in this question. Okay. So for that purpose, I have basically put in a few illustrations along with this question. The first one, as you can see, is complementation, right? So what is complementation? Sometimes what happens is that two different genotypes, they will like individually, if there was this particular mutation, right, wherein there was this capital A and small b, individually, if this would have been present in one of the flies, it would have given a white eye color, right? On the contrary, there was this other mutation wherein the A was homozygous recessive, but the B was homozygous dominant. It would also have given a white eye color in this other fly, right? But sometimes what happens is that when two different mutations from these organisms come together, what happens is now this A is heterozygous and this B is also heterozygous, right? So as a result of that, it will basically lead to a red eye color, right? The molecular level aspect is shown in this illustration over here, right? That there might have been two enzymes that might have been involved in the pigmentation of the eye. Over here, this is a defect in one of those enzymes. This is a defect in the other enzyme, maybe, right? So when we have a heterozygous condition, that is two different mutations are coming together to basically nullify the mutation, right? The eye color is wild type or dominant over here. It is getting a red eye color, right? So the mutation is getting nullified. Such a process is known as complementation, right? Now coming on to uniparental disomy. So this is also something that we have already discussed. It is basically a mode of inheritance, right? You can always consider uniparental disomy as a mode of inheritance because in your lectures you have studied it along with this concept known as genomic imprinting. It might have confused you, but like I what you need to understand is uniparental disomy is a kind of inheritance. Sometimes this kind of inheritance could lead to certain disorders which are genomic imprinting disorders, right? But these are two separate conditions. So uniparental disomy basically means that you are basically receiving uh, two sets of chromosomes from one particular parent, right? So uniparental is one parent and disomy means two sets, right? So over here, how it could occur is basically that you have a normal gamete, right? During its mitosis, there could have been an error due to which one of this, there could have been a non-disjunction of chromosomes, right? Due to which there would have been monosomy in one of the cells and a trisomy in the other cells, right? When after this the chromosomal, the sister chromatids are formed, what happens is it will break into two daughter chromosomes, right? The telokinesis and telophase will happen, then karyokinesis, cytokinesis, these things will take place. So it will basically break into two daughter cells, and what will happen is that one of the daughter cells will be normal. Right. Oh, okay. It will not be normal because instead of having just one of this and one of this, it has basically two sets of both these, right? So it will be a tetrasomy, which is not viable in humans. Tetrasomy is viable in some plants, some animals also. It is not viable in humans. So this will basically be washed out, right? Now, this is a normal looking chromosome because once if the copy numbers are checked, it will have the appropriate copy number, right? But what the defect is over here is basically that both these chromosomes or homologous chromosomes are of maternal origin, right? It did not get this blue colored chromosome that was coming from the father. It basically got both the sets from the mother. So it is a maternal isodisomy, right? What would have happened with this monosomy individual? It monosomic would have been non-viable, but sometimes the error correcting mechanism or the proofreading mechanism, what it does is that it will basically help this to get duplicated. Sorry, this will basically get duplicated so that this can be viable and it can survive. In this case, what is possible is that one of the individuals gets this, the other individual does after cytokinesis and karyokinesis does not get any chromosome. Okay, so like this was tetrasomy because there were four sets. This will be nullisomy because is no set at all, right? While this particular individual, which again looks normal, it has two homologous chromosomes, but both are from paternal origin. So this will be paternal isodisomy. This is how uniparental disomy basically occurs. Then, okay, we have to basically discuss genomic imprinting, right? So, just like I said earlier, what is genomic imprinting? What will happen is that there will be this gene, right? For example, this is the female and this is the male, right? The female, the male. What will happen over here? Both will form mRNAs, but in imprinted genes, what happens is, for example, there is maternal imprinting. What will happen? It will silence the paternal chromosome, right? It will silence the paternal chromosome and only, I think this is written incorrectly. Yeah, this should be other way around. Okay, in maternal imprinting, what will happen is that the paternal chromosome will be silenced and only the maternal one will express. On the contrary, in paternal imprinting, the maternal will be silenced and only the paternal will express, right? And how does this silencing take place? These black dots, these represent methylation. Okay, so there will be certain level of methylation through which this silencing will occur, right? Due to which this particular genome will be allele will be silent and only the other one will express, right? Then what is anticipation? Anticipation, like I have already discussed this uh, with respect to anticipation. So what happens is that in certain disorders which have repeats in them, right? For example, Huntington's disorder has CAG repeats, trinucleotide repeat, which is CAG, right? In these disorders, what happens is that with successive generations, the number of repeats increases. And if the number of repeats will increase, then the disease will come at an early age. Maybe this was at an age of 50. This came at an age of 40 and this came at an age of 33, right? So the age of onset will also reduce, right? And the severity of the disease will increase because the number of repeats is increasing, more toxic protein is being formed. So the severity will increase, right? So such a basically concept is known as anticipation. Okay. So please read this question. I'll just get back to you. Okay. I'm sorry for the interruptions, just because of my throat. Okay. So there is this other question which says, huh, it says that a process marked as A in the image given below, which results in the generation of recombined chromosomes from paternal and maternal chromosomes, is known as. What is does this process signify? Wherein maternal and paternal chromosomes will basically be recombined. So is it allelic recombination? Is it non-allelic recombination? Is it allelic duplication or is it non-homologous end joining? Which one of the following is it? So moving on to the solution, it will basically be allelic recombination, right? How? Because recombination over here is a process by which pieces of DNA are broken and recombined to produce new combinations of alleles. And such crossovers will result in recombination and the exchange of genetic material between maternal and paternal chromosomes. As a result, offspring can have different combinations of genes than their parents. Why is it allelic recombination? Because basically the alleles are aligning, right? So it is allelic recombination. How would it have been a non-allelic recombination if basically the maternal one was present? Okay. Uh, Okay, we are keeping the maternal one as this only, and instead of paternal, instead of this thing, what would have been present over here? It would have been maybe this arrow over here, then this region, and then this arrow, right? So this particular allele would have aligned with this particular allele, right? These are two different alleles. So that is how it would have been non-allelic recombination. And as a result of this, it might have resulted in allelic duplication. How? Because instead of these two sets, one of these arms would maybe have four sets, the other might have had one set, right? So it would have resulted in allelic duplication, which is not the case over here. Also, it is not non-homologous end joining. You will understand when it is an illustration for non-homologous end joining. It is something that is not even being talked about over here. Right? So this is a final question for this particular assignment, which is basically knockout mice models are best suited to study which one of the following: haploinsufficiency, loss of function mutations, recessive disorders, or all of these. So again, this being a simple question, I'll jump to the solution, which is basically all of these. So I think it is pretty self-explanatory. I do not need to explain how basically knockout mice models are able to basically study all of these concepts, right? So that brings us to the end of the first assignment for today. Now, I'll be moving on to my next slide. Let me just stop presenting. Okay, right. So, let me see if I can share that from here. Okay, yeah, I have the option. Let me just share it. Okay, sorry. And I have not really modified this. I'll do it before uploading the lecture. Right over here, we will basically be studying certain concepts which I saw that people face difficulty with, specifically on the discussion forum or on the personal mails that I have gotten. So, in order to explain those particular concepts, I will basically go through this presentation. So, the first concept that I have mentioned over here is basically this loss of function mutation and gain of function mutation, right? So, let me just get the pointer to explain better. Huh. So what is loss of function mutation? As the name suggests, that there will be a gene or a mutation which will lead to the loss of the original function of the gene, right? Gain of function means that the gene might gain an additional function, or it might basically if it was a protein coding gene, it will maybe code more protein, or maybe it will code a different protein also along with the original function, right? So any such mutation will be a gain of function mutation. So I have tried to put in these illustrations to explain you the loss of function and gain of function mutation with respect to cancer. Right, so cancer is a very hot topic, and I thought that this is one of the examples wherein both these mutations have to occur in order for somebody to actually get cancer, right? So that is why it was a good example in my perspective to explain these two mutations. So first, I need to, I actually briefly described this in the previous class also, but since that was past my time of explanation, so I would go through it uh, again. So, first, we will just see the illustration on the left, right? So it says normal genes and mutated genes, right? And it says it uses two terms. One are tumor suppressor genes and the other are proto-oncogenes, right? So tumor suppressor genes, as their name suggests, are basically genes which will suppress the tumor formation, right? So over here, they have given this analogy that it will basically work as a brake, right? And proto-oncogenes is basically the gene that has the tendency or the ability to develop into a cancer causing gene, but proto means it is in a silent state, right? If at all it will get mutated, it will develop into oncogene, which is basically a cancer gene, right? So for cancer to occur, if the tissue is healthy, then your tumor suppressor genes will be working and your proto oncogenes will not be working, right? So the accelerator over here will remain off and the brake will always remain on, right? But if the both these genes are mutated, then you will have cancer, right? Which means that the accelerator is basically working at its maximum and the brake is basically removed, right? So such a situation will cause cancer. Let us see it over here. What happens in normal genes? There might be a gene that would regulate cell growth, right? Then, if there is one mutation in that gene, over here they say that the oncogene becomes active, right? Earlier it was inactive, that is why it was known as proto-oncogene. Now the oncogene has become active. So, it will cause some changes maybe in the cell, but the tumor suppressor genes are still functioning, which means that there will not be a development of cancer completely. Like it will basically act as a protective mechanism so that this organism who has activated an oncogene will not develop cancer, right? So this person will be susceptible to develop cancer because out of two things, one has already gone wrong, but the other thing will be protecting this person. Then what happens is sometimes due to some mutations, this tumor suppressor gene will also go bad, right? So what will happen? This car has an active accelerator. It is accelerating and the brake is lost, right? So what will happen? It will go and collide. And such a collision over here is a homology to cancer, right? So in cancer, what happens is there is loss of function of tumor suppressor genes, and there is gain of function of proto-oncogenes, right? So, I hope it helped you understand both loss of function mutation and gain of function mutation, along with how actually cancer develops in a very layman term. Okay, because different kinds of cancer have different mutations, different pathologies also. But this is the general understanding of cancer that we have, right? So, somebody also talked about mRNA proofreading mechanisms in the discussion forum. And that person basically asked that if there is proofreading of mRNA or if there is mRNA surveillance, then how is it possible that basically mutations occur? So, how do mutations basically occur is that obviously there will be proofreading, right? And polymerases will very frequently actually make errors, right? But mutations don't occur that frequently. Most of these errors will be basically proofread and they will be corrected. But it is not always possible for any kind of surveillance to basically be 100% accurate, right? So because it is not 100% accurate, this proofreading mechanism, maybe one or two mutations they get uh, kind of, just a moment, one or two mutations might get missed, due to which basically mutations actually occur, right? So it is not about how can mutations occur if there is a surveillance or proofreading, because mutations, while replication or while even transcription, there are a lot of errors that is made by our enzymes. Okay, but there are a lot of other mechanisms that are keeping these in check, so they will rectify these errors, or if they are not rectifiable, then they will just tag these particular mRNAs or these particular DNA strands for ubiquitination or for digestion or degradation, right? But sometimes these might not work as efficiently, they may go bad, right? And one or two mutations might be missed, which is sometimes enough to cause maybe sickle cell anemia. That is just one missense mutation, right? So, just these mechanisms might not be enough all times, at all times, right? So, I have given you two illustrations just to understand what mRNA proofreading is. I will make these slides available by uploading it on that Google documents. I hope you people have access to that because I upload it every week. Okay. So this was just one question that I wanted to answer because I thought that others also might have similar doubts. Okay, then this I mentioned in my previous discussion that I wanted to actually include a slide on DNA slippage, but I forgot it that time. So I have included it today. So I just wanted you people to understand how this trinucleotide repeat expansion would actually occur. Right. So sometimes what happens is what are trinucleotide repeats? It is basically a repeat where there are three nucleotides and multiple of these three nucleotides are present, right? Maybe this okay. Maybe this C. Oh, why is it not working? Maybe the CAG, which is one trinucleotide raised to power 10 is present, is normally present, right? Up till 35 of these are actually normally tolerable in humans. Otherwise, if it increases that, then Huntington's is a possible pathology that you will basically face, right? For example, some individual has a CAG, 10 repeats of the CAG, right? How does it happen that his or her progeny might have CAG repeats Maybe 15. It is basically possible due to this particular uh, what phenomena which is known as DNA slippage. So in DNA slippage, what happens is a moment, please. Okay. So in this, how will this DNA slippage basically occur is I'm deliberately trying to speak a little slowly because I think that might help with the throat. Okay, and I hope I'm audible. If I'm not, I'll speak at my normal tone again. So what happens in this DNA slippage is basically that maybe one of these conditions will arise, right? Maybe in this strand, this loop will be formed due to this slip out, right? It is known as a slip out. Sometimes why this slip out occurs is because I don't know if my video is visible. So if I show it using my hand gestures, whether it will be visible or not. So maybe I'll stop presenting for a while so that you people can see me, right? Because I'm not aware if otherwise you can see me or not. Sometimes what will happen is, yeah, here, do you want to ask something? Okay. Anyway, sometimes what will happen is that maybe this is one strand and this is a new strand synthesis that is going on, right? So what will happen is that because there are so many CAG repeats, this synthesis was going on, going on, going on, and at some point, this DNA polymerase fell off, right? Now it had to be basically recruited back to this particular position. Now, when it fell off this DNA strand, it was at this trinucleotide repeat position, right? From here to here is the trinucleotide repeats. So it was at this position, maybe over here when it fell off, right? And what will happen when it has to be recruited again? Maybe it cannot recognize at which repeat it was when it fell off, right? And maybe it fell off from here, but when it had to be recruited, it got recruited over here again. So this portion, it got synthesized twice, right? So the repeat basically expanded. Or sometimes there could be another possibility that could occur, which is basically that since these are CAG repeats, right? So there is C and G, a lot of it over here. And we know that C and G, they form triple bonds between them, right? So sometimes what happens is that there will be self bonding between these, right? So when the DNA gets unfolded for replication, sometimes a loop might still remain between this DNA, right? The other is basically linear, but there is this loop that is remaining because there is a lot of CG interaction between them, right? So there was not proper unfolding of this DNA, and maybe when the polymerase was synthesizing, it synthesized this portion twice, right? So that is another possible explanation of how DNA slippage could occur. So let me just get back to my presentation. Hmm. So over here, as I have shown, there are these three possibilities over here. Wait, uh, these slip out possibilities. It could either be a heteroduplex on this chain, a heteroduplex on this chain, or it could also be homoduplexes on both the chains, right? How could it have formed? Maybe the DNA. Just a moment, I cannot find my cursor. Maybe there was a DNA damage, right? When there was a DNA damage, maybe, right? For example, if you just consider this particular part was lost. So this particular part had to be folded, right? That is why this occurred. or maybe during recombination there was the slippage that I just explained, or such a slippage could have occurred during transcription maybe, or maybe while unwinding the DNA there was some problem with the unwinding due to which such a condition arose, right? So these are the possibilities due to which a DNA slippage might occur, and it might cause trinucleotide repeat expansions, right? So these are certain, this is a depiction of certain detection techniques for triple nucleotide expansion. You do not really need to go into the details of this depiction. What you can just see is the techniques that have been mentioned. So the linkage analysis was the one that was brought in first, and it was considered as a gold standard technique up till around a decade earlier, right? After that, microarray was brought in, which was also used to detect this trinucleotide repeats. Along with this, fish was also seldom used, but microarray and fish, these usually had some limitations and errors in their data. Then the latest techniques that are being used is the NGS sequencing and the long read sequencing, right? So these are very efficient techniques and they are being used at present for the detection of trinucleotide repeat expansions, right? So moving forward, there was this, although I have discussed anticipation a couple of times now, but I also came across this doubt wherein somebody actually had a doubt between anticipation and penetrance, right? Ki whether anticipation, I don't really remember that doubt. It is present on the discussion forum actually. Maybe you can go and read it. I am not sure what exactly the statement was, but what I gathered out of that statement was that is penetrance. Uh, I really don't remember. Okay, anyway, this slide I've included over here just to explain that anticipation and penetrance are two different, basically, phenomena. In case of Huntington's, right, anticipation is basically again the severity of the disease is increasing and the age of onset of the disease is decreasing, just like I explained while we were discussing the previous year's solution question paper, right? So that is what anticipation is. I hope I don't need to explain it again in such a limited time span. What is penetrance? Penetrance is basically that if you have the phenotype for the disease, will it express or not, right? So in case of Huntington's disease, what happens is that if you have 27 to 35 CAG repeats, it is still a normal case, right? Up to 35 repeats, it is a normal case, and you have 0% likelihood of developing the disease phenotype, right? But if you have more than 35 CAG repeats and less than about 40, right? Then the penetrance is basically reduced. Means that you might have this condition which is basically more trinucleotide repeats, but you might still not be expressing the disease, right? So there is incomplete penetrance when the repeat numbers are between 36 to 40, or it is less than 42 also. Somewhere in literature, I have also read that uh, 241 also if people have the CAG repeats, it shows incomplete penetrance. So some people might not express the disease altogether, right? But according to this particular illustration, if a person has more than 40 repeats, right, the person will definitely show symptoms for Huntington's disease. The symptoms might be less severe in case that person has 40 repeats, and it might be much more severe if they have more than 56 repeats, but they will definitely show the disease symptoms, right? So 100% penetrance above 40 CAG repeats is present in patients with Huntington's disease. So I hope it is clear to you people. Moving on to translocations, right? So this explanation basically stems from a question that was asked on the discussion forum, which basically said that uh, this though you might have studied in SIR's course, know that translocations are one of the most common chromosomal aberrations that occur, right? So in that particular question, what was asked was that uh, one, why do translocations occur so much? And two, why are some translocations seen so continuously? Why is it not random? Right? For example, we talk about Philadelphia chromosome, in which translocation occurs between chromosome 9 and chromosome 22, right? So why is that a thing? Why is that translocation much more common than some other kind of translocation, right? So the explanation for this thing basically lies in this particular line, which says that translocations basically occur when two chromosomes break. Chromosome breaks share time and space, right? So the two chromosomes that are breaking apart, they should be temporally and spatially aligned. So within a nucleus, it might be possible that some of the chromosomes, they by default lie closer to one another than some other chromosomes, right? So there might be more chances of translocations that are seen in these two chromosomes than in different chromosomes. Also, for translocations to occur, there have to be similar breaks on both these different chromosomes, right? So if these similar breaks have to occur, they have to have similar sites maybe, right? For whatever, for example, some enzyme is causing a break, right? So that enzyme should be able to cause a break in both these chromosomes at sites which are very similar. So some of these chromosomes will have identical sites, the others might not have such identical sites, right? Which is how these translocation basically aberrations, they are basically temporally and spatially very uh, dependent on these two criteria, time and space, right? So this is a normal translocation that is occurring in this particular illustration on the left. In this, if the correct ends are aligned, then correct chromosomes will basically form. But if incorrect ends are aligned, then there will be a translocation that occurs, right? And along with this, sometimes on right, I have said that sometimes there is joining of correct ends is blocked by some reagents, right? So it will basically favor joining of incorrect ends, due to which some translocations are more common than others, right? Also, there is another aspect to it which is possible, which is basically that not all kinds of translocations are tolerable in humans, right? There might be some translocation which, if it occurs at all, the cell won't survive, right? So that translocation will not be tolerable, and thus there will be that cell will die, and you will never be able to actually see that translocation to occur. It might be lethal for the cell, right? So that is another aspect of why certain translocations are seen more often than others, right? Moving on to the next slide, and just a moment. Okay. So this slide discusses candidate gene selection, which was one of the major query points in the discussion forum. So what is basically candidate gene selection? It means that there are certain genes when you physically or genomically map a disease, then there will be certain uh, genes or certain regions in a genome which will more frequently differ in the people that are actually having the disease than the normal population, right? So when such a thing happens, then those particular regions will be our candidate regions, right? That because these are differing in the affected population than the normal population, maybe this is the place where which controls this particular trait, or this is the particular place where a particular mutation might have occurred. So that is how candidate gene selection works, right? For example, in this illustration, they have basically mapped that there is a trait. It is basically involving these particular genes, which are A, B, C, and D, in different ratios. These genes are involved in actually phenotypically giving this trait, right? So maybe if there is a mutation in any one of these genes, it would affect this trait at various degrees. For example, A and D are affecting this trait, but they are not affecting to a very major extent, right? Because there is a curve, like the curves going like this, right? It is not, it is affecting, it is not affecting on a very major level. So maybe the trait is not that severe, right? Maybe if there is a defect in B or C, the trait will be more severely affected, right? Because the amount of contribution that these two regions are having on this particular trait is more as compared to A and D. And maybe if there was a defect in E and F region, the trait is not getting affected at all, because anyway, these two regions are not involved in phenotypic expression of this trait, right? So I hope what basically in a layman's term, what candidate gene is, this you have understood. Now, from a paper which was published on Trends in Genetics, I have basically pulled out this particular illustration which says how basically the candidate gene selection will occur. So they will first basically design a study which will contain the case individuals or the affected individuals and a control group or the healthy individuals, right? Then they will basically define phenotypes for these groups. Then candidate gene and identification and candidate variant selection will occur between these genes, after which they will basically study haplotype analysis and linkage disequilibrium. These are two terms that I will discuss further in my lecture. Then they will basically go for the estimation of sample size and statistical significance of their findings, after which they will basically analyze their exploratory data, and there will be quality control of genotyping data. So it will be verified on multiple levels, after which there will be testing statistical association, right? There will be more statistical studies on that to find out whether whatever data they have received is by chance or it is actually valid, right? Then they will do some functional validation assays to basically finally prove their hypothesis that yeah, whatever gene candidate gene they had selected, whether or not it is actually involved in the disease, right? So that is how this works. Okay. So just like I said, haplotype, I will be mentioning what haplotype is. So first, let us go through its definition, which I have picked up from genome.gov. So a haplotype is basically a physical grouping of genomic variants or polymorphisms that tend to be inherited together, right? So it will not be just a sequence of DNA or a sequence of base pairs or nucleotides that are continuously present. It will basically be a physically put together grouping of single nucleotide polymorphisms, right? Which will basically represent a unique reflect a unique combination of variants that reside near each other on a chromosome, right? So these haplotypes, like it has been depicted very nicely in this particular illustration. You can make out that these are particular polymorphisms, and when we group these polymorphisms present on these particular number bases, for example 2 and 6 and maybe 12, 12th position of this base, right? When they are grouped together, they basically form haplotypes. And why are we grouping them together? Because these haplotypes are more or less inherited together, right? And in diseased, these basically differ in diseased individuals than they do in the healthy individuals, right? So that is why they are grouped together over here. So moving forward, I will just need another. Okay. So haplotype, just like I mentioned, the more often than not, they are basically passed on as an entire group to the progeny, right? So they are inherited together. So, for example, in this particular illustration, if you see this generation 4, let me just switch to a pen. If you see this generation, this is generation 1, this is generation 2, 3, and 4. So if you just see this individual 8 and this individual 6, right? And what does this sign basically signify? It is basically a consanguineous marriage, right? So marriage between cousins. So in these two individuals, both had this haplotype, which is signified in pink, which when they mated, some of their children got only this haplotype from both of them, right? So when basically genomic analysis was done, this haplotype came up that these affected individuals had basically double copy of this particular haplotype. That is why they were affected. So just like we talked in gene inheritance, these particular people do it is kind of a recessive disorder, right? Because their parents are not affected, they are carriers, but the children are affected, right? So it is kind of a recessive disorder, which how did they actually find out that what particular region was basically involved in this disorder? By studying these single nucleotide polymorphism patterns in these children and in their parents, right? So it is more or less self-explanatory because I have extensively basically dealt with the pedigree problems, right? So that is why I don't feel the need myself to explain a pedigree properly. But if you have any doubt, you can ask. In that case, I'll explain it, right? Then moving on to dynamic mutation. So I have just mentioned the Wikipedia definition of what a dynamic mutation is, because somebody again had a doubt about dynamic mutations and whether or not these mutations could be any other mutation other than a trinucleotide repeat could also be called a dynamic mutation. This was their doubt, if I'm not wrong. So, according to the definition on Wikipedia, what dynamic mutations are is that in genetics, a dynamic mutation is an unstable heritable element where the probability of expression of a mutant phenotype is a function of the number of copies of the mutation, right? So, where will number of copies be a problem? It will be a problem where repeat expansion is involved, right? So, not any kind of mutation can be a dynamic mutation. It will just be certain types of mutations, right? For example, trinucleotide repeat expansion. Then another person had a doubt in what exactly these repeats were, right? So it is actually a very valid question. We can just not assume that everybody is aware of what repeats is. So just to explain that, I wanted to put in this very basic slide, which basically says that what are repeats in genetics, right? So it is a very basic slide. I am not sure if everybody is aware of all the basically terms that are used over here. And most of it you don't even have to worry about, at least for this course. But if you are interested, you might go and read about these terms, right? So what are repetitive DNA sequences? So in the DNA, there are certain naturally occurring sequences which are repetitive in nature, right? It will not always be A, T, G, C, C, G, C, 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 T, A, something like that. Okay. Sometimes naturally it will occur as ATG, 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 CCC, and so on, right? So there will be repetitive sequences that are present in DNA, that are present in that is present in the genome naturally. So these are basically repeats that are mentioned in terms of genetics, right? These repetitive DNA sequences, they can either be tandem repeats or dispersed repeats. So what is dispersed repeats? First, let us deal with this part because I will not go into much detail in this part. So dispersed repeats, as the name suggests, they are dispersed, right? Means they are present at different locations in the chromosome. But what will happen is that maybe a particular sequence is present at different locations in a genome, right? Maybe it is present from nucleotide number, base pair number 10 to 58, then it is present, 10 to maybe 25, then it is present at 35 to 50, then it might be present at 115 to 130. So it is present at different, the same repeat is present at different locations in the chromosome, in the genome, right? So in that case, it will be known as a dispersed repeat, right? Transposons are examples of dispersed repeats. So retrotransposons, as the name suggests, are RNA-like transposons. DNA transposons are again DNA transposons. I will not really go into the explanation of what transposons are or what these retro or DNA transposons are, because there's a separate classification for this also. And for that, basically, I will have to explain what transposons are, which is a very huge topic. So maybe you can just read about it, right? It will not be asked in this particular course, but for your knowledge, maybe you can ponder a little about it, right? Then these were dispersed repeats. Now, also transposons are also known as jumping genes. So you might as well search it using that term also, right? Let me write it over here so that I'm sure that you've understood. Okay. Transposons are also known as basically jumping genes. Okay. All right. So the other class over here is basically tandem repeats. So tandem repeats are repeats that are present one after the other. For example, VNTR people study in molecular biology, right? Variable number of tandem repeats. So these are repeats that are present one after the other, like the example that I mentioned, ATG, 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 right? So these repeats, on the basis of how many repeats are there, they are divided into satellites, mini satellites, and microsatellites. So there is no consensus as to how many repeats, at how many repeats will it be called a mini satellite, at how many repeats it will be called a microsatellite. There is no consensus on that, but just for reference, maybe you can understand that micro means very small, so it will basically be 6 to 10 repeats, maybe maximum, right? Mini satellites might range up to 100 repeats, right? 100 base pair repeats, right? Not 100 repeats, 100 base pair repeats. So a sequence of 100 base pairs will be repeated again, right? That might be mini satellites, and similarly, satellites will be even bigger, right? So I hope repeats is also understood what repeats are. Then I wanted to discuss the coding versus non-coding region of the genome. Also, I feel that I will not be able to do the assignment number 4. I don't know why I aim so much. I always think I'll be able to tackle more in every given class, but I am not. I'm sorry for that. But maybe I'll just be able to finish this particular lecture, right? Also, my throat is not helping me with it either. So I'm so sorry for everything. Okay. Coding versus non-coding region of the genome. So these are two representations that I have basically mentioned, which both signify that what percentage of the genome is basically coding. So in the representation on the left, you can see that the portion colored in black is basically the protein coding region, right? And the one in gray is the non-coding region. The one in white is the untranscribed region. So in Escherichia coli or E. coli, 88% of its genome is protein coding. In the yeast, right? Saccharomyces cerevisiae, 68% of its genome is coding. In C. elegans, 24% of its genome is coding. And in Homo sapiens, only 2% of the genome is coding, right? For in us, only about 2%. It is an approximate, only about 2% of our entire genome is protein coding region, right? So over here, what you can see is from bacteria to yeast to basically C. elegans to humans. The complexity of the organism is increasing, but the protein coding region is decreasing, right? So it is basically kind of an inverse effect. So there is another representation on your right over here, which basically again lists that Homo sapiens have only 1.4% region that is protein coding. Drosophila have around 20% region that is protein coding. C. elegans have around 25% region. Around Bidapsis thaliana, which is a plant which is very widely used for genetic analysis, has about 29% of its region that is protein coding. And as we move towards basically prokaryotes, the protein coding region increases, right? From top to bottom, as the complexity decreases, the protein coding region basically increases. So, this is something that you can keep in mind, and you can also always you will ask a question as to why the protein coding region has gone down so much, right? Also, let me just give you a reflection of how many protein coding genes are present, right? For example, in Arabidopsis, around 27,000 genes are protein coding, right? In C. elegans, it has around 20,000 genes. Homo sapiens have around 25 to 30,000 genes, right? Mus musculus has more number of genes than humans. So this mouse, the lab mouse that we use, they have more number of genes than us, right? So they have around 30,000 genes. So I just want to through these two basically slides, all I wanted to tell you was that there is not a direct correlation between the number of genes and the complexity of the organism, right? So humans, these many number of genes only contribute 2% of its entire genome, right? But still, it is the most complex organism. So this basically tells us how important this non-coding region and the untranscribed region must be, right? So obviously it does. These regions, the one in gray and white, they obviously do help humans because these might be regions that have enhancers or they have different kinds of transcription activators, enhancers, then promoter regions. All these are present in these regions, which basically aid this 2% of the genome to function to its fullest, right? Then another point that I wanted to discuss was genomic versus physical mapping. So it is just a very simple illustration of what a genetic mapping or genomic mapping is and what a physical mapping is. So in genetic mapping, there will be certain markers that people will have in mind that, okay, if this is the particular gene that we want to deal with, then or this is a particular gene that we want to deal with, then this is a particular marker that we can use for it because they are present very close by on the chromosome. This we know because of our chromosomal study or our genome-wide association studies. These are certain things that are already in our knowledge, right? Just a moment. Yeah. Okay. So these are things that are already in our knowledge. And what is physical mapping? In physical mapping, the segments will be broken down into multiple fragments, and then, depending on the overlap, they will basically align these fragments and find out that, okay, this particular genetic marker lies in close proximity to this particular gene, and so on, right? So, out of these two, one important thing that you should keep in your mind is basically which one of these techniques is more accurate. So, what do you think? Will it be genetic mapping or will it be physical mapping? Okay, let me answer. It is basically physical mapping. This is a more accurate technique. Let me show you another illustration. This one, right? So, as the title suggests, there are discrepancies at times between genetic and physical maps, right? So for a same genomic region, or over here for a chromosome number 3 for East, if you draw a physical map versus if you draw a genetic map, there will be certain discrepancies that would arise, right? For example, over here, CHA1 is the first gene that is present and GLK1 is the second one. On the contrary, in the genetic mapping analysis, what they found was GLK1 was the first and CHA1 was the second. Similarly, you can see other such discrepancies also over here. A little bit position difference is always going to remain, but such major discrepancies is the one worrisome thing, right? So in this case, which one will you basically trust? Physical mapping or genetic mapping? Basically, you will trust the result of physical mapping, right? So genetic mapping data may have limited resolution, right? If the progeny is not enough. So if the sample size is not large enough, then the resolution will go down for genetic mapping. Also, it may have limited accuracy. Why is its accuracy going to be limited? Because we are not breaking the fragments, right? If we are not breaking the fragments, then as we know, there are certain combinatorial hotspots or cold spots. Hotspots are certain locations where recombination occurs very frequently. Cold spots are certain regions which might be linked, right? There will be linkage disequilibrium and recombination will not basically occur that frequently, right? So there is no absolute relationship between genetic and physical distances, but out of these two results, you should basically trust a physical map because if you read it in literature, it will basically favor physical mapping more, right? You can understand why by reading more literature. Okay, then there is this illustration for transgenic versus knock-in or knockout mice, right? So this sir has already discussed in his slide. I just wanted to go over this concept once again in very brief, which is basically what is the difference between a transgenic and a knock-in or a knockout, right? In transgenic, what will happen is that there will be a recombinant DNA that we will be taking, and we will directly inject it into whatever recipient in at the pronuclear stage, right? So at the pronuclear stage itself, we will basically inject this DNA into the recipient, and then this particular fertilized egg will be injected into a foster mother who will basically and this particular embryo will develop into a transgenic mouse. We are talking in case of a mouse model, right? In knock-in or knockout, what will happen is that we will take a vector, we will target a gene through homologous recombination in a petri dish. So this will be an in vitro and culture. Then this targeted embryonic cells will be selected and expanded, right? So there will be certain selection processes of the cells that actually could be successfully transfected using this targeting vector. And then these selected cells will be basically injected at the into the early embryonic stage. Over here, blastocyst stage is shown. So in this particular stage, these targeted embryonic stem cells will be injected, right? And this embryo will be basically again put into a foster mother, and it will basically create a knock-in or a knockout mice, right? Also, there were certain variations in the knockout condition. For example, it could have been a constitutive knockout. Knockout basically means that the gene is not going to function now, right? What does constitutive knockout means? That it is definitely not going to function for its entire life, right? For example, if this was a wild type mouse, it had this particular gene which was functional, and this gene of interest was basically knocked out, so this will gene will have no expression at all. Then there could be conditional knockouts, right? So this conditional knockouts could either be tissue specific knockout or inducible knockout, right? Why do we need a conditional knockout? We need a conditional knockout because sometimes if this gene is not at all functioning in this mice, right, it might not develop into this bigger mice, right? It might be embryonic lethal. At the embryonic stage only, this mice would not survive if this gene was required for development, right? Or some major part of development, right? So in these cases, we need tissue-specific knockout. Also, the same genes might perform different functions in different organs or tissues in an organism, right? So over here, why are we using a tissue-specific knockout mouse? Because maybe this particular gene would have been required for the growth of the mouse. Also, even in an adult mouse, if we just turn this gene off, it still might be lethal for the mouse. For example, we can say that there is one gene which is involved in muscle regulation, right? But we only want to target it in this particular tail muscle, in the tail or the leg muscles, right? Or the arm muscle. We don't want it to affect the heart muscle, right? So what we'll do is a tissue specific knockout, in which what will happen is that the promoter that we will be using will be tissue specific. For example, for this particular gene, there will be a separate promoter, maybe there will be a separate promoter for its arms and legs, and a separate promoter for if this particular gene was present in its heart, right? So we will just use the promoter or induce the non-expression of this gene through basically stimulating the promoter that was present in the arms and legs and not the one in the heart. So this is one kind of conditional knockout. The other kind of conditional knockout is inducible knockout mouse, right? For example, in this, uh, okay, you can go back and you can read this particular thing. It will help you understand inducible knockout. Uh, I did not put a slide for this over here. TET on, TET off system. Okay, so TET over here is for tetracycline, right? So TET on, TET off system you can read for inducible knockout mouse. In this, what happens is that you will basically externally give this mouse something to eat or drink or something, due to which it will affect the expression or the function of this gene, right? If you suppose if you are giving tetracycline in some cases, it will basically lead to a non-expression of this gene, right? So it is inducible because through diet or through water or through some external injection, you are basically inducing the knockout of this gene in a full grown mice, right? In a full grown mice. So, okay, another thing that I wanted to discuss was basically double cloning, double selection in cloning, which basically says this. Okay, I hope I'm back. The power got disconnected. That is why, yeah, this also got disconnected. Let me share my screen again. Okay, so I will just finish with this presentation. Then I'll just leave you guys. So let me go through it very quickly. So, for example, there was an origin of replication in a vector, and there were two selectable markers, which I mentioned as selectable marker 1 and selectable marker 2. And then there was this region, which is colored in green, which is basically what we are trying to put inside this is basically our gene of interest, right? Let me just type it as gene of interest. Okay, sorry. This is my gene of interest, right? So, what happens in a double selection is basically, suppose this origin of replication was from here, right? There was this selectable marker, and this was happening, and it would only replicate up till here, right? So it would mean that if the gene of interest has been transferred to whatever clone it had to be transferred, to whatever host it had to be transferred, it will basically contain this particular selectable marker, which would basically, it could be some kind of antibiotic resistance, for example, neomycin resistance, right? So if this gene of interest would have been cloned over here, it would contain neomycin resistance. So if we grow it in a Petri plate, what will we find? And we put neomycin in this dish. So only the resistant population will grow, and this resistant population will also have a gene of interest, right? If we also, along with this step, we also need to check that this part we don't want, right? We want the replication to just end over here where it should end. So for that, we will basically add some kind of different marker over here. For example, it could be a toxin. So when this particular gene of interest is basically cloned over here, after neomycin resistance, if these particular cells which did show neomycin resistance, if they would have this other marker, right, that is a toxin. If the substrate for this toxin to function was given, then the cells expressing this toxin will die because obviously it will be toxic for the cell, right? That is why it is named toxin. It will be toxic for the cell. That is why the cell will die. So it will ensure that the vectors that have regions beyond this point will not get cloned, right? So they might have the gene of interest, but if they also have the toxin, they will just not get cloned. So this is basically double selection. I actually wanted to put a better explanation, illustration over here. So if in case anybody had any difficulty in understanding, I will make I did not get a suitable illustration. I will make an illustration and I'll just represent it in my upcoming week's lecture, right? So let us move forward. Okay, so that was actually my last slide. So at least this slide I finished well in time. Uh, sharp at 5 today. So there's no real point in starting the fourth week's assignment, which I did have prepared. But since your time is up and there is another class that I know is from 5 to 7, right? My colleague Muchandra will be taking that class. So maybe you guys can join that class. We'll discuss week four's assignment in week four itself, along with the extra concepts and topics for that particular week. If you have any doubts, you can post it in the discussion forum. The TA for the course, which is not me, she basically takes care of answering your doubts on the discussion forum. I personally also always go through that discussion forum to basically prepare this concept slide so that if there is some such question that you guys are facing, I that is my only source of information, right? So I basically look at that and I try to incorporate those concepts in my slide so that it might help you guys. But you can also write to me personally. That is always an option. Just do not spam me. You can refer the first week's video for my mail ID, right? And that is going to be it for today. You guys can leave. I'll stay back for a while because I need to save the recording, but you guys can leave. So thank you so much. I'll just end the recording. Okay.